The structure of DNA is arranged in a double helix,
which is also anti-parallel.
The monomers of DNA are nucleotides. The nucleotides are made up of a phosphate, deoxyribose, and a nitrogenous base. There are 4 types of nitrogenous bases, which are called adenine, which pairs with thymine, guanine, which pairs with cytosine.
Okay, so we're going to be talking about transcription. And so what's going to happen is there's going to be an RNA polymerase, which is being drawn right now. And the RNA polymerase catalyzes the DNA and pries it apart whilst hooking together the RNA nucleotides. This turns the DNA into RNA, which follows the same base pairing rules as DNA, except that in RNA, uracil is the substitute for thymine.
This process is ended by a termination sequence sent by the RNA polymerase to end this coding process. When RNA is first composed, the coding is not always lined up properly. The introns, or extra unwanted nucleotides, are spliced out so that the RNA strand codes properly,
which leaves the extrons.
The smaller mRNA strands that were created through transcription can now fit into ribosomes. The ribosomes read the code of the RNA and pairs it with
anticodons on the tRNA.
Can now release and go into cells to form proteins.
For the entire process to start and end, the ribosome needs to read the start and stop codon.
Amino acid chains are folded to create the final protein structure by the primary structure, secondary structure, tertiary structure, and quaternary structure. The primary structure is a linear sequence of amino acids, shown as AA, and they are held together by a peptide bond to create a polypeptide chain. The secondary structure refers to the way the primary structure starts to fold upon itself. There are 2 ways in which we can visualize this. There is the alpha helix, which looks like a coiled structure. This has hydrogen bonds that run up and down it, and it stabilizes the coiled structure, or the alpha helix. The second way it can be represented is the antiparallel or the beta pleated sheet. It looks like this.
And you have the amino N, which is the N, and the carboxyl N, which is the C, which line up with each other. Third stage is the tertiary stage, and it's the higher order of folding in a polypeptide chain. It depends on distant group interactions. One of those distant group interactions is called hydrophobic packing. So this is a protein, and it's surrounded by water, and on the inside you would find the hydrophobic group, and it's on the inside because it does not like to interact with water. Now on the outside you have the polar groups, and they are there because they love water. So after the tertiary structure, there's the quaternary structure, and that refers to the bonding between multiple polypeptides. So right now I'm drawing a folded up polypeptide, and I'm drawing 4 of them. And this structure is the interaction between the 4 polypeptides, and each is turned into a subunit. A protein that is properly folded up is called conformation, and that means that every step that I'm writing here and that I just explained must be correct in order for conformation to happen. Why is protein important? Protein is important because if it is misfolded, then it doesn't work. And this leads to many neurological diseases such as Alzheimer's, Parkinson's, and ALS. Also, protein is used to build cartilage, hair, nails, skin, and blood. Thank you.
Thank you.
Can we stop it?